Today we are going to discuss about complement system. Complement system. What happens in the complement system? How it is helpful? Okay, complement system is seen in both innate immunity and adaptive immunity. So, what is the use of this complement system? They help in defensing. Defensing. How they'll defense? They help in defensing against microorganisms. Microorganisms. Okay, microorganisms. What happens here? Normally, complement proteins will be present. These proteins are inactive. Normally they are in inactive stage. Okay, they have to be activated. This activation is done through different processes. We'll go through further. Okay, normally these complement proteins are numbered from C1 to C9. Okay, they are numbered from C1 to C9. From C1 to C9, we have a critical step. That critical step is C3. Okay, the activation of C3 is very important. So, this activation is a critical step. Activation of C3 is critical, very important. Okay, this C3 activation or a cleavage of C3 is done with the help of by three pathways. Okay, it is done with the help of three pathways. What are those three pathways? One is classical pathway. Second one is alternative pathway. Third one is lectin pathway. Okay, so we'll discuss each pathway in detail. First we'll see classical pathway. Okay, what happens here in classical pathway? Normally whenever a microorganism enters into the body, they will evoke a response for formation of antibodies. Okay, this antigens will evoke a response where formation of antibodies will occur, and these two antigen antibody will combine together and they will form a complex called as antigen antibody complex. Whenever this complex is formed, okay, this complex will bind to C1. Okay, they will bind with the C1. Whenever they are binding with the C1, they will stimulate. Okay, they will stimulate C1. Okay, they will stimulate C1. So what for what they have to stimulate C1? For protease activity. Stimulate C1 for protease activity. Okay, protease activity. What happens here? By activation of C1, this C1 will act on C4 and this will act on C2. By acting on C4, this C4 is proteolized into C4A and C4B, and this C1 will also act on C2, causing proteolysis of C2 into C2A and C2B. What will happen here? This C4B and C2A will combine together and they will form a complex, and this complex is called as C3 convertase. C3 convertase. Okay, this C3 convertase has protease activity which causes splitting of C3 into C3A and C3B. Okay, C3A and C3B. Now C3B will bind with C4B and C2A. Okay, they will form a complex. Okay, C3B. Okay, we'll write like this: C4B, C2A, and C3BB. This complex is called as C5 convertase. Okay, C5 convertase. What this C5 convertase will do, as every convertase is having protease activity, this C5 will convert C5 into C5A and C5B. Okay, this C5B will combine with other complement factors C5, C6, C7, C8 and C9. They will combinedly form a complex called as MAC. This MAC is called as membrane attack complex. Membrane attack complex. So, what is the use of this membrane attack complex? This membrane attack complex will act on the bacterial cell wall. If this is a bacterial cell wall, whenever this membrane attack complex is attached, they will form a pore on the bacterial cell wall. After formation of pore, this is pore. Whenever pore is formed, they will cause cell lysis. Whenever there is a cell lysis, the cell will die, and this remnants of cell lysis is digested, or digested means they have to be phagocytosed by macrophages. This is how classical pathway will kill bacteria. Okay, next, in this formation we have seen C3 and C3A and C5A. They will act as a chemoattractants. Okay, chemoattractants. So what is the function of these chemoattractants? They will recruit leukocytes. Where they are recruited? To site of infection. What they will do? Evoke inflammatory response. Okay, next we will see lectin pathway. We have a complement protein called as MBP. What is this MBP? This is called as mannose binding lectin, MBL, or it can be called as mannose binding protein. Okay, this MBP has different name: mannose binding lectin, or it can be called as mannose binding protein. What they will do, this protein will bind with the polysaccharide. Where do you see this polysaccharide? This polysaccharide is present on bacterial cell wall. Okay, so normally bacteria will contain lipopolysaccharide. Okay, lipid and polysaccharide. In that polysaccharide, if mannose is the major content, this mannose binding protein will bind with that polysaccharide. Okay, with that mannose. Okay, so where do you see this type of lipopolysaccharide? You can see it on gram positive bacteria, viruses, some viruses, not all viruses, some viruses, okay, and you can see in some fungi, okay, fungi. This lipopolysaccharide, okay, this LPS is present, lipopolysaccharide is present on gram positive bacteria and in some viruses and some fungi. This LPS, this lipopolysaccharide will contain mannose as one of the part, and this mannose, whenever this type of bacteria enters into the body, then mannose binding proteins are activated and they will bind with this bacteria and they will form a complex. This MBP plus LPS, okay, they will form a complex and they cause C4, activation of C4. This C4 undergoes autoproteolysis. This autoproteolysis will cause splitting of C4 into C4A and C4B. And this C4 is can also split C2 into C2A and C2B. Then both C4B and C2A will combine together and they will form a complex called as C3 convertase. C3 convertase. Okay. C3 convertase. And this C3 convertase will act on C3 and they will cause splitting of C3 into C3A and C3B. Okay, C3 convertase will convert C3 into C3A and C3B. Now C3B will combine with C4B and C2A and they will form a complex called as C5 convertase. C5 convertase, which will convert C5 into C5A and C5B. C5A and C5B. This will combine with C6, C7, C8 and C9 and all together they will form membrane attack complex and the process repeats as same as classical pathway. Okay, this is about lectin pathway. What is happening here? Some bacteria will have lipopolysaccharide. In that lipopolysaccharide, mannose is a component. This mannose is identified by mannose binding protein. So they will combine with that and they will cause activation of C4. This activation of C4 causes autoproteolysis of C4 and it causes splitting C4 into C4A and C4B, C2 into C2A and C2B. This C4B and C2A is combinedly called as C3 convertase, which helps in conversion of C3 to C3A and C3B. This C3B will combine with C2A and C4B combination and they will form C5 convertase, which will convert C5 into C5A and C5B. This C5B will combine with C6, C7, C8 and C9 and they will form a complex called as membrane attack complex and the process is same as classical pathway. The difference is between classical and lectin is classical and lectin pathway. What is the difference between these two? In classical pathway, the activation is through C1. Here, where antigen and antibody complexes are acting on the C1 and causing activation of complement system. Here, directly bacteria is involving, which is having a lipopolysaccharide. It is combining with MBP. Okay, MBP, and they are activating C4. C1 is not involved in lectin pathway. It directly starts from C4. In classical pathway, it will start from C1. That is the difference between classical pathway and lectin pathway. Next pathway is alternative pathway. Alternative pathway. Okay. In alternative pathway, directly C3 is activated. C3 is activated directly. C3 is activated by chemical hydrolysis. Chemical hydrolysis. Okay, chemical hydrolysis. What happens here in the alternative pathway? C3 is converted into C3A and C3B. What happens here? This C3B will combine with factor B and they will form a complex called as C3BB. Okay. This is acted upon by factor D, which will split C3B into BB and BA. Okay, this C3BB has C5 convertase activity. C5 convertase. It is called as a C5 convertase. What it will do? It will convert C5 into C5A and C5B. Okay, C5 convertase. And C5B will form a complex with C6, C7, C8 and C9. Okay, together they are called as membrane attack complex. Okay, so if you see classical pathway, lectin pathway and alternative pathway, here it is acting through C1 plus antigen antibody. Here MBP plus bacteria, they are directly acting on C4. Okay, here in alternate pathway there is chemical hydrolysis of C3. So in classical pathway C1 is involved, in lectin pathway directly C4 is involved, in alternative pathway directly C3 is involved. In all these three pathways C3 is common. C3 is common pathway and C5. After C3 is activated, C5. Okay, and formation of membrane attack complex is common in all these three. Okay, this is about classical pathway, lectin pathway and alternative pathway. And we will see the biological functions of complement system. Biological functions of complement system. Okay, complement system. So what happens? They'll cause cell lysis by forming membrane attack complex. What this membrane attack complex will do? They'll cause pore formation on bacterial cell wall. Okay, like this they'll kill the bacteria. After killing, the remnants are digested by macrophages. Next, they will have vascular phenomena. Vascular phenomena. What is this vascular phenomena? We have seen C3A and C5A are released whenever C3 is converted by C3 convertase and C5 is converted into C5A and C5B with the help of C5 convertase. And we have seen C4A also. This is least important. Okay, least important. But these two, C3A and C5A, are very important. Okay, what they will do? They act on mast cells. By acting on mast cells, they causes release of histamine. They causes release of histamine from mast cells. Histamine from mast cells. Whenever histamine is released, the histamine will cause increased vascular permeability. How they will increase vascular permeability? By vasodilation. Okay, this is how they'll invoke an inflammatory response. And what is the function of C3A and C5A? They act as anaphylotoxins. Okay, they act as an anaphylotoxins. Next, the third function is C5A will activate lipoxygenase pathway. On what uh, lipid they will act? Essential fatty acid. They'll act on arachidonic acid. Okay, and this lipoxygenase pathway is activated in monocytes and neutrophils. Okay, monocytes and neutrophils, and they causes release of prostaglandins and prostacyclins. Okay, next, fourth function is C5a is a powerful chemo attractant. Okay, they will attract neutrophils, monocytes, eosinophils, and basophils. Okay, basophils. Next, C3b. This C3b will bind with bacterial cell wall. Okay, they'll act as opsonins. Act as opsonin. By binding with the bacterial cell wall, they act as an opsonin. By attachment of C3b causes phagocytosis. They act as opsonins, and they will attract neutrophils and macrophages. Okay, this C3b will act as an opsonin, so that they will attract neutrophils and macrophages to the inflammatory site or infection site, and they cause phagocytosis. So this C3b will act as a sugar coat on the bacterial cell wall, so that this sugar coat, this C3b is like a sugar coat which will attract neutrophils and macrophages, which causes phagocytosis. So this is about complement system, and in next part we will see clinical point of complement system. Okay, in different diseases, how this complement system is causing action. Okay, we will see different clinical scenarios.